Hello, welcome to botany class students. Today we are going to see the dark reactions in the photosynthesis. Okay, so dark reactions, where these dark reactions will occur, what is meant by dark reactions? That's the one what we will get in that is a simple and the direct question in the NEET exam, what we'll say. Okay, dark reactions means when the chemical based reactions, when the enzymatic reactions occur in the chloroplast of the plant cell, those are called as the dark reactions. So these dark reactions will occur in the stroma of the chloroplast. Okay, so there the light intensity will be taken up and they will do the photosynthesis in the presence of the water molecules and taking up the carbon dioxide from the outside atmosphere. And here the end product in the dark reactions is nothing but the glucose molecule, which is further converted into the sucrose and the starch as the resolved food material in the plant body. So how these dark reactions will occur, and in all the plants, same type of dark reactions, and we are saying the light energy from where the plants will get the light energy. That's the one what here we have to see. So here for this, we can go with the slides what I have prepared already. Okay, so here I have given the key points and the explanation what I will give. With that only we can easily go for the question and answers in this chapter, in this topic itself. So in the dark reactions, these dark reactions are nothing but the enzymatic reactions, enzymatic chemical reactions will occur, which are completely related with the otherwise done with the enzymes that are present in the stroma of the chloroplast. That is the enzymatic reactions. And these enzymes will be present in a large number, there is a large quantity according to the species to species where they are growing in the stroma of the chloroplast. That is a semi-solid liquid that is present in the chloroplast. And this is completely the energy consuming process. In the light reactions, the energy is going to be released in the form of the ATPs and the NADPH2s. But that energy will be consumed again in the dark reaction. So it is an energy consuming process for the plant body. But here the stored food material will be in a starch molecules which can be stored for a long period of time and which can be reutilized at any time by the plant body for all its mechanical purposes. Okay, so that's the one what here we are saying the energy is consumed. Okay, food is prepared and energy is consumed in this form, in this. And here, in these plants, again, the dark reactions will be done in the three different manner that means three different types of dark reactions can be seen based on the plants, how they will perform the photosynthesis and where they were growing. Based on these 2 points, again, the dark reactions are of 3 types. The first one is the Melvin-Kelvin cycle, RC3 cycle. Okay, only the Kelvin cycle also they can say. Melvin-Kelvin, Kelvin cycle, RC3 cycle. And the second one is the CAM cycle. Okay, second one is the C4 cycle. And the third one is the CAM cycle. C4 cycle means here only in the xerophytic adopted plants. The plants which will show the xerophytic adaptations, those will do the photosynthesis with the C4 cycle. Okay, and this is not the pathway, this is a cycle, because all the way the first substance is going to be regenerated. So it's not a pathway, it's the nothing but the cycle, what we have to remember for the competitive exam. And the last one is the CAM cycle. CAM cycle means the crustacean acid mechanism, what we can say. So in this where this CAM cycle occurs, in which type of plants? CAM cycle will occur only in the true xerophytes, the plants which are growing in the xeric conditions. Then only we will see the CAM cycle is going to be occurred. Now comes the first one is the C3 cycle. C3 cycle occurs in the plants, in the mesophytic plants, and in the maximum plants, C3 cycle of the photosynthesis occurs. Here, why we are saying it as a C3 cycle, otherwise the Melvin-Kelvin cycle, otherwise the Kelvin cycle. So this was the first invented by Melvin-Kelvin. And again, we can call it as the Kelvin cycle also. Usually, we will call it as Kelvin cycle, otherwise the C3 cycle. C3 cycle word came based on the first stable compound that is formed in this reaction, this cycle. That is a 3-carbon compound that is called as the phosphoglyceric acid PGA. Based on this, the cycle got the name as the C3 cycle. And again, this C3 cycle will be done in 3 different steps. That is the carboxylation phase, reduction phase, and the regeneration phase. In the carboxylation phase, only one step will occur. They will ask this also. Okay, only one step will occur, that is where the absorption of the carbon dioxide and fixing of the carbon dioxide occurs. And in this carboxylation means 2 different steps will be done. That is a carbon fixation and the oxidation process. Both will be occur, and this both reactions will be done by the only one enzyme, that is the Rubisco enzyme. This is the enzyme which is abundant in the outside atmosphere. Okay, much more amount of the Rubisco can be seen in the outside atmosphere. That is the Rubisco enzyme. And this Rubisco enzyme will start this process. That is the first compound which is absorbing this carbon dioxide, and in the presence of this enzyme, what it will do. So what is the substance? The substance is the ribulose 1,5-bisphosphate, that is RUBP. And this is going to be carboxylated by absorbing the carbon dioxide molecules. See, if we are taking the 6 RUBPs, then 6 carbon dioxides are taken up and the 6 water molecules will be taken up. Then in the presence of the Rubisco enzyme, it is carboxylated into the stable compound, that is the 3-phosphoglyceric acid. And this 3-phosphoglyceric acid will be going into the reduction phase, where 2 steps will be there. That is the in between, the intermediate step will be there. So the end product of the reduction phase is the glyceraldehyde 3-phosphate, G3P, what we will say. And this G3P will be the one which is going to be making a pool-like manner. And these 12 G3Ps are present in a pool, and in this the one will be going into the cytosol for the preparation of the glucose, otherwise the sucrose molecule. And the rest of the 5 in the out of 12, if one goes, that means 2. Okay, they are present in a pool. In the pool means 6 in a chain. Okay, multiples of the 6 will be placed for its convenience in the pool of the G3P. So in that first 6 G3P that are present in a row, those in that is one will goes into the cytosol for the synthesis of the sucrose, otherwise the glucose molecule. Then the second row will be present. If 12 are there, multiples of 6 means here 6, here 6. So one will goes means from the second row also one more. That means total 2 G3Ps will go into the cytosol for the preparation of the 1 hexose sugar, otherwise 1 glucose molecule or 1 sucrose molecule. Okay, usually that is the glucose molecule only going to be formed. And the remaining ones are in the first row 5 G3Ps, and in the second row 5, total 10 G3Ps. Now these 10 G3Ps will do the regeneration process. That is, they will participate in the regeneration process. And here in the regeneration process, what they will ask, how many enzymes are utilized twice? 2 enzymes will be utilized twice. That is the aldolase and the transketolase enzyme, which are going to be utilized twice in the regeneration phase. And how many molecules of the xylulose 5-phosphate are going to be formed? Okay, 2 times the xylulose 5-phosphate are formed in each and every time. 2 molecules. That means total 4 molecules of the xylulose 5-phosphate is going to be formed. And all these are going to be regenerating into the ribulose 1,5-bisphosphate. And here, how many times the isomerization occurs and how many times the dephosphorylation occurs, that is the phosphate group is going to be released. Like that also they will ask. Just go through the cycle once again. Okay, and then in this one, the last what they can ask us, okay, that is the if the one glucose molecule has to be formed in a one by this C3 cycle, then how many carbon dioxides are needed? That is the C6H12O6 is the formula for the glucose. That means 6 carbons are needed. The C6 means 6 carbons. So in that 6 carbon dioxides need to be absorbed to form one glucose molecule. That means 6 times the Kelvin cycle has to go thoroughly so that one glucose molecule is going to be formed. That is the process in this when in this entire step. Okay, so here what we will say, this one is going to be they can ask in the word problems. That means if 3 carbon dioxides that is the basic one we have to say here. Okay, if 3 carbon dioxides are absorbed, then here 6 ATPs and 4 NADPH2s are going to be NADPH2s. This is also okay. Just correct it. So 6 ATPs and 4 NADPH2s, and these 4 NADPH, and then this one is in the 3 multiple forms. That is the 3 table. Okay, and if 6 carbon dioxides are absorbed, means then 18 molecules of the 6 ATPs. Now that is 18 molecules of the ATPs and the 12 molecules of NADPH2s are going to be taken up. Like that, we need to say the word problems. They will ask us in this manner only in the word problems. The next one is the C4 cycle. In the C4 cycle occurs in the plants which will show the xerophytic adaptation in them. So those plants only perform the C4 cycle. So here, in the C4 cycle, stomatal apparatus are completely restricted on the lower epidermis, and then this one is also present in the sunken type, and the chloroplasts are present in 2 different forms. That is the chloroplast dimorphism can be seen, and then here in these 2 types of the chloroplast, the dark reaction C4 cycle is going to be occurred. That's the one what we have to say. In this Rubisco enzyme is a place. The one cycle will be done in one chloroplast. Rubisco enzyme will be present. In another chloroplast, Rubisco enzyme is not present. So they will go with the enzyme PEP carboxylase enzyme. So here the first compound which absorbs carbon dioxide is the PEP, phosphoenol pyruvic acid. And this phosphoenol pyruvic, and this one, again, we have to say, and here PEP carboxylase is the enzyme which is carboxylated the phosphoenol pyruvic acid into the oxaloacetic acid, which is a 4-carbon compound, the first stable compound. Based on that, the cycle got the name as the C4 cycle. And in this, what we have to say, the Rubisco enzyme is not sufficiently present in the mesophyll chloroplast. So here this process starts from the mesophyll tissue of the leaf body only. Okay, so here the mesophyll chloroplast does not have the Rubisco enzyme, so they will start with the PEP carboxylase. And in this one can start the photosynthesis when the carbon dioxide concentrations are less in the outside atmosphere also. That is the one what is the most important one in this. Okay, and they can start this process in the less availability of water and less carbon dioxide concentrations. And in this also, in this only, C4 cycle and the C3 cycle both can be seen in the plant body. So that means much more amount of the food is going to be prepared by this process. That's the most important one. And where the C3 cycle occurs, in the bundle sheath chloroplast, C3 cycle will occur. And this C3 cycle will be having, starting with the presence of the enzyme Rubisco. And this Rubisco enzyme is the one which is having the 2 enzymatic groups in it. Okay, one is for the carbon dioxide, another one is for the oxygen. And in this, these 2 substrates, carbon dioxide and oxygen, acts like the competitive inhibitors. So which will come first and blocks the enzymatic group. Based on that, the reactions will be done by the Rubisco enzyme. Okay, and in this C4 plants, the bundle sheath cells usually should be the colorless, but here they are having the green color because of the presence of the very big oval shaped chloroplast in them. So those are the type of arrangement of the chloroplast in the bundle sheet cells is called as the Kranz anatomy. Okay, that is looks like a garland-like occurrence. Next comes the CAM cycle. CAM cycle means crustacean acid mechanism, which is going to be seen only in the true xerophytes. And in this one, this process occurs in two different timings in the plant body. That is, the first one starts in the dark in the night, during the night time, and the second one that is occurs in the daytime. That is, in the night time, they will do the photosynthesis by absorbing the carbon dioxide, and the conversion of the phosphoenol pyruvic acid into the oxaloacetic acid and the malic acid will occur. And during the daytime, what will happen? Complete malic acid is converted into phosphopyruvic acid, till that only. Like that, the process will be occurring. And again, in the night time, they gain this pyruvic acid converted into phosphoenol pyruvic acid. Then the next steps will be continued. That like that only the process will be done. That is the CAM cycle. Okay, crustacean plants only we will see. Like this, they will ask, and this is the daytime and the nighttime. Entire steps will be similar to the C4 cycle. Okay, right? And if you are having any doubts, you can share the doubts in the comment column, and don't forget to subscribe this channel. Right? Thank you.